In the name of God. Can you hear me?
Yes.
Yes.
Hello, Dr. Faizabadi. How are you?
Thank you very much.
Can we start?
Yes, of course.
Thank you. Okay, it is Ehsan Mostafavi from Pasteur Institute of Iran. I would like to welcome you for joining our meeting for the panel of emerging and re-emerging infectious disease. First of all, I would like to thank Professor Faizabadi and Professor Abdullahi who were arranging this Congress, the International Congress of Microbiology. Then I would like to thank our participants of the meeting. And finally, I would like to thank our speakers who accepted our invitation to join for this panel. Today we have 4 presentations from different areas of microbiology. First of all, we will have presentation from Professor Mathieu Picardeau from Pasteur Institute of Paris. Then we will go to Dr. Lucia Moro from Center for Tropical Diseases in Italy. And then we will continue with Professor Katja from Institute of Bacterial Infectious Diseases in Germany. And the last one will presentation of Professor Mohammad Mehdi Gouya, who is the director of Center for Communicable Diseases. I'm sure that we will have very good presentations and good discussions during the meeting. For each of the speakers, we will have almost 15 to 20 minutes speaking, and then we will continue with 5 minutes discussion and questions. If you agree, we can start with Dr. Picardeau, who is the director of Department of Biology in Biology of Spirochetes in Pasteur Institute of Paris. He is the director of Center for WHO Collaborative Center for Leptospirosis. Hello, Picardeau.
Hello, everybody.
Hello. Floor is yours, please.
Okay, thank you. Thank you very much for your invitation, and thank you for inviting us. So I thank all the Iranian Society for Microbiology for this Congress, and it's for me a great opportunity to present what we are doing here at the Pasteur Institute on a neglected disease. So I will talk today about leptospirosis. So I'm going to share my screen. Okay, here.
Okay.
So can you see my presentation full screen?
Yes, yes.
Okay.
Thank you.
So today I'm going to talk about the agent of leptospirosis. So I'm going to talk about what we are doing here at the Pasteur Institute here in Paris. So I will first make an introduction about spirochetes in general, and then I will focus more on leptospira and leptospirosis. So as you probably know, leptospira belong to a large family of bacteria called spirochetes. And among these spirochetes, so you have major human and animal pathogens such as Borrelia burgdorferi, the agent of Lyme disease, Treponema pallidum, the agent of syphilis, and leptospira, the agent of leptospirosis. As you can see here on this slide, so all these bacteria, they share a common morphology. So all these spirochetes are long and thin helical bacteria. And so for leptospirosis, so we have there was a paper a few years ago by WHO estimating to more than 1 million severe cases of leptospirosis per year and 60,000 deaths due to leptospirosis worldwide. As you can see here on this map, so incidence is much higher in Southeast Asia, but also we have high incidence in America, in the Caribbean islands. And as you can see here, there's also a question mark about Africa, because there are very few studies on leptospirosis in both human and animals, and so it's not it's poorly documented. The leptospirosis in Africa is poorly documented, but the incidence should be pretty high on this continent. So leptospirosis is a zoonotic disease. So two groups of animals can carry the bacteria. So the rodents, the rats, that are the main reservoir of leptospirosis. These rodents are asymptomatic carriers, so they do not develop the disease. And you have all the other mammals, so wild animals, but also domestic animals. And these animals can develop the leptospirosis, are more or less susceptible to the disease. So all mammals can carry the bacteria in the kidney, and they're going to excrete the bacteria in their urine and then contaminate the environment. So the main route of transmission to human is an indirect route of transmission. It's the contact of the abraded skin or the mucous membrane with the water contaminated by the urine of these animals. You can also have a direct contact with the urine or the animal, but it's more anecdotic. So usually when you have a contact with the water contaminated by the urine of the animal, so the bacteria is going to spread into the organism. And so the patients can develop severe disease that can lead to death. The main problem with leptospirosis is the diagnostic. The diagnosis of leptospirosis, because there's no specific symptoms for leptospirosis. It mimics many other diseases and tropical diseases, as you can see here. So leptospirosis can mimic symptoms of dengue or malaria in tropical countries. So there are a long list of differential diagnoses with leptospirosis, with also now COVID-19, which also can have some symptoms pretty similar to leptospirosis. So as I told you, so the main route of transmission is the contact of the water contaminated by the urine of animals. So usually when we have outbreaks of leptospirosis, it's during recreational water activities or during flooding events. So usually you have water, and so you have rodents or any other mammals that are going to excrete the bacteria in their urine. And after rainfall, so all these urine or bacteria that are present in the soil are going to be resuspended by the water and going to contaminate the river. So and then you're going to have contamination or outbreaks of leptospirosis. So this is the main, the most common transmission of leptospirosis. As you can see here on this picture, so here it's Mayotte. So the Mayotte island is a very small island between Madagascar and Africa. And as you can see here, every year during the rainy season, so here you have the rainfall, and here the blue line corresponds to the number of cases. And each time during the rainy season, you have a peak of cases of leptospirosis. And this is the case in most tropical countries. So you have always a good correlation between the rainy season and outbreaks of leptospirosis. So leptospirosis is a neglected and emerging disease. So it's a neglected disease because we have limited information on the burden of the disease. As I told you, this is particularly true in Africa, for example. It affects the most neglected and marginalized populations. We lack adequate diagnostic for leptospirosis. There's no effective control measures. As I told you, the main reservoir are rats. And as you probably know, it's almost impossible to eradicate the population of rats. There's no universal vaccines available for leptospirosis, and we have a limited understanding of the epidemiology and the pathogenesis. So it's a neglected disease, but it's also an emerging disease because of the demographic and ecological changes. So today there are more than 1 billion people living in slum areas, and in these slum areas, so it's you have rat-borne transmission of leptospirosis, and we know that the population living in these urban slums is going to double in the next 25 years. It's also emerging because of the climatic changes with the global warming, and you have more and more extreme climatic events such as heavy rainfalls, flooding events that are responsible for large outbreaks worldwide. So for all these reasons, leptospirosis is an emerging disease. It's also, as I told you, a neglected disease because, as I told you, the diagnosis tests that are available today, the performance of these diagnosis tests are very poor. And so we lack information on, we lack reference laboratories for the diagnostic of this disease. And so we have very few information on the burden, and we have a misdiagnosis also because of the symptoms of leptospirosis that are mimicking many other tropical diseases. And so we have missing evidence of burden. And so there's no political advocate and little funding, etc., etc. So there's a vicious circle about leptospirosis. Just here to, as an information, so here you have the burden of leptospirosis and some other diseases such as rabies and dengue. And so here the burden is estimated by using the DALI, so the disability adjusted life years. So here you count the number of years of lives that are lost due to a disease. And you can see that the burden of leptospirosis is similar or even higher than some other diseases such as rabies or dengue, for example. So what about the agent of leptospirosis? So these bacteria are spirochetes. As you can see here, they are long and thin bacteria with a helical morphology. And they have also what is specific for spirochetes. Spirochete is the presence of endoflagella. So the flagella are not external like some other motile bacteria, but they are inside the bacteria in the periplasmic space. And so you can see that here under the dark field microscope, these bacteria are very motile, and you can see this helical-like morphology. So there are major knowledge gaps in the field. So as I told you, we have limited understanding of the epidemiology and the pathogenesis. So there are many knowledge gaps. So what are the reservoirs? So rodents are the main reservoir, but any mammals and maybe also probably some reptiles or birds can also be a reservoir of the pathogen. What is the role of the environment? So we know that these bacteria can survive in the environment in some specific conditions, but for how long? And so we don't know much about the ecology of the bacteria into the environment, and this is very important during the transmission, because as I told you, the transmission occurred usually during contact with contaminated water. So what are the drivers of the emergence? So I told about the change in the climatic change. So the appearance of more extreme climatic events such as flooding events, the fact there are more people living in urban areas, in slum areas. So we need to know better what are the drivers of this emergence worldwide. What could be the effective control measures? What method for early diagnosis of leptospirosis? Because so leptospirosis can be treated by antibiotic. Today there's no resistance to antibiotic for these pathogens, but the main concern, the main problem is the early diagnosis of leptospirosis. And the tools that we have today are not very efficient. And so there are two other questions. So what are the strains that are circulating worldwide? And this is very important to develop new diagnostic tools, for example. And what makes some species more pathogen, more virulent than other pathogens. So today I don't have much time, so I will focus more on these two different questions, two different points. So first of all, in the last few years, we isolated several strains, mostly from the environment. And we here at Pasteur, we have a sequencing platform, so we can sequence the strain from our collection and new strain that we can isolate. And so by sequencing the genome of these bacteria, so we can look at the gene contents, the genome organization. We can do phylogenetic trees, so looking at the diversity of strains that are circulating. We can do a global mapping of strains circulating worldwide. We can do a pangenome analysis, and we can also try to understand the evolution of virulence among these pathogens. So here it's a phylogenetic tree from a few years ago, from 4 years ago. So at that time, there was only 21 species in the genus Leptospira. And these 21 species were clustered in 3 different groups. So the saprophytes, so these species are bacteria that are isolated from the environment and they are non-pathogen. Then you have the pathogens that are responsible for leptospirosis in both human and animals. And for example, you will find Leptospira interrogans, which is one of the most frequently isolated species among the patients, among severe cases of leptospirosis. And you have a third group that we call at that time intermediates. And these species, and they are potentially pathogens. So there are few publications showing that some of these species can be pathogen for both human and animals. But the status of virulence of these species are still not very clear. So I told you, in the last few years, we isolated and sequenced with different, with our collaborators. We isolated several strains from different places. So in France, in Algeria, in Japan, Malaysia, New Caledonia, and Mayotte. And so we isolated more than 100 environmental strains. And so we sequenced the genome of these strains, and we compare the genome of these strains with the ones that were already available in the databases. And so we are able to identify more than 40 new species. So now the genus Leptospira contain 64 species and not 21 like a few years ago. These species, so all the new species that we have identified are the one indicated here by a black dot. So here you have a phylogenetic tree based on the core genome. So you have species that are responsible for responsible for the most severe cases of leptospirosis. So these are the ones that are here. So that are responsible for leptospirosis in both human and animals. And then we identified several new species from the environment that are belonging to this P1 subclade and to the P2 subclade that was called before intermediates. And then here you have the saprophytes with many new species that we have isolated also. So now we have this better idea of the diversity of species among the genus. We can try to understand to better understand the evolution of variants and why some species are more frequently isolated from human and animals. And so, for example, so here you have the same phylogenetic tree with the P1, which corresponds to the pathogens, the P2, the intermediates, and here you have the saprophytes. So as you can see, among the P1, only the species that are present here have been isolated from human or animals. They can be isolated from the environment, and they induce disease in animal models like hamsters or mice. So for leptospirosis, as I told you, rodents like mice and rats are asymptomatic carriers. But in this case, we look at the colonization of the kidney by this bacteria. So, and in the hamster is a susceptible model. So in this case, when you inoculate a pathogenic strain in the hamster is going to die of leptospirosis. So all the other species that we have identified that are belonging to the P1 or P2, as you can see, they are not able to induce disease in both the hamster and the mouse model, except for Listeracea, which is isolated from rats, which can induce colonization in mice. So you can see there's a clear discrimination between these species that are responsible for leptospirosis in both human and animals, and they can also induce disease in animal models, and the other ones that are present here and here. So we can classify all these 64 species in 3 different groups: the virulent pathogens that are responsible for leptospirosis in both human and animals, what we call the low virulent pathogen, because we don't know exactly what is the status of virulence of these species, and the non-pathogens here, which correspond to the saprophytes. What is important also to know is that all the diagnostic tests that we are using are targeting these species. So that's why also we don't have lots of information about the presence of these species in animals or in humans, is because, for example, for PCR, the primers and the probe that we're using for PCR are targeting these species and not this one. So that's why we don't find these species in both human and animals. So we need to develop new tools to try to identify if these species are present in leptospirosis in both human and animals. And then, so now we have this clear classification between virulent pathogens, low virulent pathogens, and non-pathogens. You can also look at the presence of some putative virulence factors. So here, for example, a few years ago, we characterized an adenosine that is very important for the adhesion of the bacteria to the host, to the cell host. And you can see that the gene encoding for this leucine-rich repeat protein is present mainly in the virulent pathogens and almost not present in other bacteria. Hemolysins are present exclusively in the virulent pathogens and not the other one. Peptidases also in a subgroup of the virulent pathogens. Mobile elements also are important, were very important during the evolution of and the emergence of pathogens, because they are much more mobile elements like transposases and prophages in the most virulent pathogens compared to the non-pathogens, etc., etc. So this is still an ongoing analysis that we are doing in the lab. And so the idea is to trying to better understand the evolution. So the idea is that the hypothesis today is that at the beginning, Leptospira the ancestor of the pathogen, were living in the environment. So they were saprophytes, and they, by different horizontal transfer and by losing some capabilities, some metabolic abilities, they adapt to a specific host and they acquired new virulence factors. So, for example, here we are very interested in trying to better understand what differentiates the virulent pathogen from the low virulent pathogen. So here there's a node of evolution that can really clearly discriminate the species responsible for leptospirosis in animals and humans from the other one for which we don't have evidence today that these species are able to cause leptospirosis. So we are using different strategies, trying to better understand what can differentiate between these two different phylogenetic groups. So in conclusion, so by isolating and sequencing several strains from the environment, but also from a historical collection, we can show that there's a great biodiversity of species among the genus Leptospira, and this is still probably still underestimated. So regularly we identify new species among the genus. There are still many question about the relevance, for example, of these environmental bacteria that are related to the pathogens. Are these intermediates or low virulent pathogens really important in public health? To answer this question, so we need to refine the current molecular diagnostic tools. What are the reservoirs of these low virulent pathogens? So they have been mostly isolated from the environment, from water, but are they present in animals? We don't know. I didn't have time to present this project today, but we also developed a core genome MLST scheme. And so we have a large database that is publicly accessible. And so we have now thousands of genomes in our public database. And so everybody can have access to this database and compare their genome to the one present in our database. And so one also of the main topic of the lab is trying to better understand what are the virulence factors, why some pathogens are more virulent than others, and how do these pathogens emerge from saprophytic strains. And with that, I would like to conclude with by thanking all the collaborators and all the members of the Biology of Spirochetes unit. If you have any question, I would be happy to answer your question. Thank you.
Thank you. Thank you, Dr. Picardeau. It was very informative, and I'm sure that our colleagues who are, most of them, they are microbiologists in Iranian society, have lots of information from your presentation. Okay, any question from the participants of the meeting?
I can unshare my presentation. Okay.
Is there any
Hello,
question?
can you hear me?
Yes, yes, Dr. Sazman, please.
Thank you so much for your presentation, Dr. Picardeau. I have a question. You talked about low virulent species and virulent pathogenic species. But again, you talked about doubts on pathogenicity of this low virulent species. Would you speak more about the pathogenicity in animals, or we're focusing on humans actually? Thank you.
So for the relevance of these low virulent pathogens, or what we called before the intermediates in terms of public health, but also in animals, today it's still not clear. So in animal models, they are not able to induce disease, but maybe we don't use the appropriate animal models. So in our models, we are using hamsters or mice. Maybe these low virulent pathogens are to show that these pathogen saprophytes are really pathogen or virulent, we may need to use another animal model. I don't know, but there are there's a few publications, and even recently there was a review on the trying to understand the prevalence of intermediates in both human and animals. So apparently there are several papers showing that we can isolate these low virulent pathogens or intermediates in animals and in humans. Usually they are responsible for mild disease. It's not severe disease. But it's still an open question. We still don't know really what is the burden of infection with these intermediates. It's still an open question, and we don't have the answer of this. It's difficult to answer this question because, as I said during my presentation, we don't have the tools to detect these intermediates. Because when you're making a PCR, for example, to detect Leptospira, the primers or the probe that we're using cannot really detect these intermediate because they are different from the pathogens, the true pathogens.
Thank you.
Thank you.
Any more question?
I have a question. So in Coxiella bonnetti, we have a single species genus, and we can only differentiate the isolates by genotyping. And we found that certain genotypes are associated with small ruminants and other are associated with large ruminants. And also, if you look at isolates from humans, you also can differentiate between acute Q fever or isolates causing chronic Q fever. And we are also not able, because they are so similar, to differentiate, is it because the bacteria are different, or is it because the host is different? The immunity of the host to the pathogen is different. So it's a very, very difficult question to answer this.
Yeah. But in the case of Leptospira, what could explain this large diversity of, because I didn't talk, so there are hundreds of Reservoirs, so there are several hundred of Reservoirs, at least more than 60, probably much more species. It's because most of these species or Reservoirs, they adapt to a specific host. As I told you, Leptospira can be carried by any mammal. And there are also some publications showing that you can isolate Leptospira from amphibians, from birds, from reptiles. So, and each time they adapt to a specific host, so you have a different genome. So they rearrange the genome to really adapt to this new reservoir.
Okay, thank you.
Thank you. Okay, I would like to thank you again, Dr. Picardeau, for your nice presentation. If you agree, we can go to the next speaker, who is Professor Lucia Moro. She is academic staff from Center for Tropical Diseases in Italy, who will present another neglected bacterial disease. Please.
Okay, thank you. I will also share the screen, just a second. And Okay, so first of all, thanks for the invitation. I'm very glad to speak today and to speak about this interesting subject, that is Trophorima whipple, that is often a neglected bacterium. So I will speak, just a second, sorry. I will speak about this bacterium today because it's the causative agent of Whipple disease. And this disease is often not recognized because the symptoms are non-specific. And the problem is if it's not diagnosticated, is not covertly treated, the prognosis of the patient with this disease is poor, and the mortality approach 100% of the case after 1 year. So a short history of the disease, that is a recent disease. It first time it was described in 1907 by George Whipple, that described a disease characterized by deposit of fat and fatty acid in the intestinal and the mesenteric lymphatic tissue. And this tissue comes from a 36 years old man that died after an history of malabsorption syndrome and chronic arthralgia. George Whipple described microorganisms in this autopsy, but he didn't imagine it was the cause of the disease, and he speaks about lipodystrophy when he described the disease. It was just many years later that after the first successful treatment with the antibiotics, it was postulated that the bacterium can be the cause of the disease. It was postulated of an infectious origin of the disease. But the bacteria was difficult to identify, so it was needed more years to identify the bacteria by electron microscopy. And just in 2001, it was possible to cultivate the bacteria, and in 2003 to complete sequences the bacterium. So the bacterium is a widespread bacterium, is a gram-positive bacteria that has a rod-shaped form that is characterized by very slow time. Okay, can you hear me, please? It's okay? Sorry, can you confirm, please? It's okay, the audio?
Yeah, it's okay.
Yes, it's okay. So the bacteria is a gram-positive bacteria that is rod-shaped and is characterized by a very slow time of replication. Actually, is lower even than mycobacteria, and is deficient in many genes that encode energy metabolism and amino acid synthesis. So it's dependent from other eukaryotic cells. The bacterium is also characterized by high diversity, high genetic diversity, and actually there's many genotypes, and type 1 and 3 are the most frequent. The bacteria is widespread, but the disease is very rare. The bacteria is widespread from human to human by oral and fecal-oral transmission, but the majority of people present just an acute infection, and they solve the infection after clearing the bacterium. Just few people unable to clear the bacterium can be persisting, colonized, or can develop a disease. And the disease can present as a localized form of the disease or a classic form of the disease, usually many years after the first contact with the bacteria. The specific deficit, immunitary deficit that provokes this inability to clear the bacterium is at the T helper 1 cell, and it provokes the inability to kill by the macrophage the bacterium inside the phagosome. So we speak also about epidemiology of multi-carriers, because in many people can be a carrier of the bacterium without developing the disease, and that actually we find the bacteria in the stool. And as you see in this slide, the prevalence of the carriage can be very different from one country to another country. And in general, the prevalence is higher in low and middle income countries and in children of this population. And because it depends also probably by hygiene condition and the kind of exposure. Actually, also inside the same family, the prevalence is higher than in the normal population. In Italy, our center performed a study investigating the presence of Troperima whipplei in the stool of our patient, and we analyzed in this study that we published in 2019 1,040 samples, and 6.9% of these samples that arrived for any reason to the hospital result positive for the bacteria. In Iran, the only epidemiological study that is available is this recent study that our colleague from Iran did with us in Italy, and that showed that there was a colonization by Troperima whipplei in immunocompromised children. Actually, it was conducted in the central Iran, in Qom, and 130 children were analyzed. The children were immunodepressed mostly because of hematological malignancy. And in the stool of these children, in 10% of the case, we found the bacteria. So about the disease, the disease is, we see, it can present as an acute and chronic disease. The acute is not the problem usually, because it's a self-resolution infection that can present as a pneumonia and a gastroenteritis or a bacteremia. And the problem is the chronic form of the disease that can be classical, and the classical form is the form that George Whipple described at the beginning of the 20th century, and a localized form of the disease. The classical form of the disease typically affects middle-aged white men, and it is also correlated to specific genetic predisposition, and especially it has been described some association with HLA and some familiar case of Whipple disease and other kind of polymorphism or variants of immune response that explain this inefficient immunity at level of the helper 1 cells. The classical form of the disease is characterized by the first phase of the disease by an intermittent altrachia and fever, so very non-specific symptom, and after some years also other symptoms develop, usually general syndrome with weight loss and malabsorption syndrome and lymphoadenopathy. But just when disease advances, in the last phase of the disease, almost every organ can be involved, but mostly the eye, heart, and the brain. And it's important to underline that the brain is often involved. More than 50% of the patients with the disease have a PCR positive for the bacteria in liquor, also without symptoms. So it's very often to have a brain involvement, even in a symptomatic phase, patient in the last phase of the disease. The patient presents at the moment of the diagnosis with joint involvement that is very frequent and general involvement. And I think it's important to put in evidence that the diarrhea, that is the symptom that very often is imaging like the typical symptom of Whipple, is not so constant, is present in 55% of the cases. In localized form of the disease, there is no gastrointestinal involvement, so the disease in localized is just one organ, one system that can be just brain, just cardiac valve, just eye, just also articular system or lymph node. And the problem is, as we say before, very often the disease is confused because, for example, with the chronic arthralgia, patients are believed to be ill by seronegative arthritis, or rheumatoid arthritis. So the patient receive immunosuppressive therapy, and with the immunosuppressive therapy, the situation of the patient became worse, and the progression also is faster. So it's important to diagnose the classic and the chronic form of the disease. For the classic form of the disease, the diagnosis is based on histology, and especially in the bacterium can be present in a patchy distribution in the duodenum, gastric and antral region, and jejunum and ileum. So because of this patchy distribution, several samples should be taken to have correct tissue for the biopsy, because if not, the risk is of a false negative biopsy. So the typical finding of in the histological samples in the biopsy is the pus positive macrophage that is typical of the disease, but that they're not so specific. So they need to be complemented by another technique, that is the immunohistochemistry, that is based on the Trophorina Whippley antibodies. But in the last decades, another important technique, that is PCR, has put in evidence its utility, and especially this important article of Fenoyard analyzed more than 4,000 samples that demonstrate the utility of this PCR that can be done in many different tissue, in many different specimens. And in this table, you can see, for example, that the positivity in saliva and stool of this PCR for Trophorina Whippley is as a high positive predictive value of the disease in selected patients when the disease is suspected. So in this paper also we suggest that they suggest that saliva and stool can be used as a screening test to select patient to which perform invasive analysis. Moreover, the PCR can be done also in other tissue, and especially it's important this technique for other tissue like cardiac valve or articular fluid, to diagnose this localized form of the disease that can present without gastrointestinal involvement. Actually, this saliva and stool screening test is very useful in the classic form of the disease, because they are very often positive, but can be negative in the localized form of the disease. So these are a scheme about how to manage the diagnosis of the Whipple disease. In the classic form of the disease, you see that we suggest to do before the PCR for non-invasive samples like saliva, blood, and stool, but later also we see the role of urine. And when a true positive test, or at least one test positive, there is a possible of the diagnosis of Whipple disease. And so an invasive sample can be performed, and for the definitive diagnosis. And also in this table, if the suspicion of Whipple disease is real, and the patient is diagnosticated of Whipple disease, is important to exclude the neurological involvement by doing the investigation by PCR on liquor before starting treatment. localized form of the disease, the diagnosis is based on true positivity. And so one positivity can be the PCR done in the organ affected by the disease, and the second positivity can be the histology and the pus staining done always in the affected tissue. And so just a few words about the treatment of the Whipple disease. The treatment disease is complex because since the beginning of the suspicion of the infection origin of the disease, many antibiotics have been proposed, but the few patients affected by the disease and the fact is rarity make very difficult to have randomized controlled trial that compare different options of treatment. So still there's no an evidence to define which is the gold standard of the treatment. The only prospective randomized controlled trial for treatment is a trial done in 2010 for initial treatment that compare at the beginning of the treatment for the patient the first 2 weeks to receive meropenem or ceftriaxone, and then both of these groups received trimethoprim and sulfamethoxazole for 1 year. So in both cases, the meropenem and the ceftriaxone demonstrate to be effective as initial treatment for this patient, and for a period of 36 months, the clinical remission was achieved in 93% of the cases. However, apart from later than after this study, some doubt about the role of cotrimoxazole in the treatment of Whipple disease presented because of intrinsic resistance of trimethoprim to the bacteria, and the frequent resistance, at least in French and German, a German strain of sulfamethoxazole. So many authors prefer to advise to use doxycycline in combination with hydroxychloroquine as a first-line treatment for Whipple disease because of the combination is bactericide, and also because the possibility of this treatment to cross the blood barrier that is important in neurological Whipple disease. The treatment with doxycycline and hydroxychloroquine in combination should be continued for 1 year, and later the doxycycline should be kept for 1 year more. This is the article that speak about the frequent relapse with the treatment of cotrimoxazole, and actually 29 patients were analyzed were compared the treatment of 29 patients receiving different antibiotics, and 14 patients receiving cotrimoxazole presented a relapse, and so the treatment demonstrated not efficient. And while in the group with doxycycline and hydroxychloroquine there was no relapse, and so is the reason why the authors advise this treatment as the first-line treatment. About the follow-up of the disease is characterized by a clinical improvement very fast in the few weeks after starting the good correct treatment. So the patient improved fast. But the problem is that the susceptibility to the bacteria remain, and so it's important to continue the treatment for many years, and actually many authors suggest lifelong. And for the follow-up, it can be very useful to check the PCR, because usually they become negative also very quickly, and so can be very useful to confirm the good answer to the treatment. And in a patient who receive who have received immunosuppressive therapy, another problem has to be considered, that is the immunoreconstitution inflammatory syndrome, as in other infectious disease, can complicate the evolution of the patient that is correctly treated with the antibiotic and need specific treatment. So thanks you for your attention, and a special thanks to Dr. Anna Beltrame and Dr. Saber Esmaili. Please feel free to ask questions if you have. Thank you.
And thank you, Dr. Moro. Thank you for your nice presentation. Any question from the participants, please?
Dr. Mostafavi, we have one question in the chat box. Please check it.
Yes, please. I cannot find the question.
Yeah.
Oh, it's private. I cannot find the question. Yes, yes, please.
Why this bacteria is more widespread in male than women?
The difference is in the disease. The disease is more frequent in the men, but the colonization is no different actually. So probably there is a genetic predisposition in men that is more frequent than in women, but it's not the colonization different, is the disease, and in particular the classical form of the disease.
So the prevalence of the infection is the same in both
Yes.
genders, but the disease is different.
Exactly.
Yes.
And very often, the typical patient that is described to have classical Whipple disease is a man and is Caucasian, but I think much study are needed to understand if in African or Asian country where the disease is less described and less frequent, it's possible that there is other form of the disease that are not diagnosticated or not investigated. So, and also because usually the disease need years for to present. So it's not usually in young people, it's in 50, 60 years old patient.
Thank you. Any more questions? Okay, I would like to thank Professor Moro from Center for Tropical Diseases who joined us, and we had a very good presentation from her. If you agree, we can go to the third speaker. Thank you. Who is Dr. Katja Mertens from Friedrich Lupfer Institute in Germany. She will speak to us about Coxiella burnet, please.
Okay, thank you. I think you see not the right. There was.
It is.
I think there's something where I can switch, right? No.
Okay.
Yeah. Did you see my PowerPoint? No.
We can see your screen now. Okay.
So which one?
We are waiting for you to share your screen.
Now.
So which one do you see? Do you see my presentation in full screen?
No, it is not in full screen. Please make it in full. Yes, now it is okay.
Okay. So I will talk to you about Coxiella bonnetti and
okay. Yes, it is now shared.
Okay, so
Yes.
about Coxiella bonnetti, the epidemiological situation we have in Europe and in Germany especially, and how we want to improve the diagnostics. So first I would like to introduce Coxiella bonnetti as the etiological agent of Q fever. Coxiella is closest relative is Legionella pneumophila. It's also a pathogen. Coxiella is an obligate intracellular microaerophilic bacterium. It is sensitive against oxidative stress to a certain extent. And since it's intracellular, which you see here, we have the Hoek stain for the host nucleus. This is a mouse fibroblast. And in green, we stain the Coxiella, which sits inside or replicates inside the phagolysosome component. It is exposed to oxidative stress. Coxiella and every Coxiella containing material you have to handle under BSL-3 conditions. Coxiella exhibit a strong tropism for the female reproductive organs, where it replicates in the trophoblast layer and may cause a necrotizing placentitis. There are two antigenic variants known from Coxiella, which corresponds to the LPS. Phase 1 bacteria express a full-length smooth LPS, and phase 2 bacteria have a severe reduced or truncated LPS, and they are avirulent in rodent infection models. This phase 2 variant only appears in the laboratory after highly frequently passaging in embryonated hen's eggs. And every isolate you find in nature will have a full-length LPS. Q fever or Crary fever is a disease, a zoonotic disease distributed worldwide, except in New Zealand and Antarctica. The bacteria are transmitted by aerosol transmission, and also ticks are discussed as vectors. The main reservoir for human infections are ruminants, especially the small ruminants, sheep and goats. An infection is mainly asymptomatic, which makes diagnosis difficult, but it can also cause a placentitis, abortion, or weak offspring. The bacteria are shed via the milk, birth products, urine, and feces. Also, cattle can be a reservoir, and here also the infection is asymptomatic or it's associated with mastitis. But also in cattle, Coxiella can cause reproductive disorders, and in cattle, the bacteria are mainly shed within the milk. And it's shown here for a goat, the cycle of infection, inhalation of bacteria. Then if the animal is pregnant, they will replicate within the trophoblast layer, and during parturition, they will be shed in tremendous amounts with birth products. These birth products can contain 10 to the 9 bacteria per gram tissue, which is a really, really high load. And if these birth products are kept unattended in the environment, they can dry up and be transmitted with the wind over several kilometers. And for humans, it was calculated that less than 10 bacteria are infectious by inhalation. So this is a major problem, especially in rural areas. Within the within the EU, we have fluctuating numbers of cases of Q fever in sheep, goats, and cattle. And this is because serological investigations or investigations at all are only done after clinical symptoms appear. And so far, we have no harmonized surveillance scheme for animals within the EU. This will be changing because we have a new animal health law in Europe, and now harmonized surveillance systems hopefully will be applied. So these data are only descriptive for the ruminants. In Germany, Q fever is a reportable disease, and the numbers within the last 5 years, as you see here, are more or less stable. This map shows you the cases we had during the last year in small and large ruminants. And this map shows you the cases reported over the last 10 years. And you can see that we have 3 hotspots in southern Germany, western and northern Germany. There are some areas where we have high frequency of Q fevers cases in animals every year. Q fever in humans is a disease normally or mostly caused by small ruminants. This is one case we had. It was a farmer's market, and these apparently healthy asymptomatic sheep gave birth to a kitten, but this Eve was infected, so all the surrounding people got infected with Q fever. The zero prevalence for Q fever in large and small ruminants is between 50 to 80% in all Europe. And we record about 250 human cases per year in Germany. The largest ever reported outbreak for Q fever happened in the Netherlands from 2007 to 2009. And it was caused by intensive goat farming. This outbreak over 2 years, there were over 3,500 confirmed acute Q fever cases in humans and over 70 Q fever associated deaths in humans. This shows how Q fever or Coxiella infection can be a really problem for the human health system, because this is associated with a lot of costs and following up costs for the health system. And also countermeasures are expensive, and they choose to eliminate the infected animals. So also this is a lot of costs, and it's for the people who must do this, it's also a mental problem. Then we have one outbreak situation which happened here in Jena, where the FLI is located. We had a apparently healthy sheep herd, which was grazing across this urban area. And after lambing season started, there were some Q fever cases in humans reported. And at the end, this herd was positive for Q fever. And in this very dry and hot summer, the wind was breezing from this grazing area across this residential area. And with increasing distance, the cases of Q fever went down. So this is a good example how environmental conditions also affect the spreading of Q fever or the transmission of Q fever from animals to humans. What are we doing in the laboratory? If we get a sample, you can do PCR on samples like milk or afterbirth material. We do not use blood because we rarely or never find Coxiella in the blood. Then you can isolate the bacteria by tissue culture cells. You can use embryonated hen's eggs, or you can also use actinic media under certain atmospheric conditions to isolate the bacteria. This is a direct detection of the bacteria, and then you can do indirect detection by ELISA. The serology which is used for veterinarian medicine is not phase specific. I explained to you at the beginning that you have two antigenic variants, the long LPS and the short LPS. And in human medicine, you can detect different antibodies against these variants. And this gives you an idea if the infection is at the onset, or if it is an acute or chronic infection of Q fever. You see this what type of antibody you see against which antigenic phase. So we want to improve diagnostics of Q fever because the serological detection, the first antibodies appear after 2 to 4 weeks after infection in animals. And then we do have shadow animals which are serological negative, which we cannot identify, and they keep the infection cycle up. Then with the regular serological test systems, they were shown that they have a gap in detection of certain types of antibodies. And then there is the antigen which is used for the ELISAs is a whole cell extract, which is done by proliferation of the bacteria in embryonated hens eggs under BSS3 condition. So you have a whole protein extract or whole bacterial extract, which might be different from each batch to batch. So we have one approach where we want to use proteins which are immunoreactive from the literature, or which we identify by our own experiments. And from these proteins, we predict B-cell epitopes, which are small peptides. And we will analyze these peptides we generate by field sera, which are positive or negative against Q fever. And at the end, we want to create a peptide array for highly specific and sensitive detection of Q fever. From our literature approach, we found over 160 proteins which are reported as immunoreactive in the literature, but only in 19 publications. And each protein was only cited once or maximum twice in a publication. Localization of all these cited proteins is either unknown, or the majority was in the cytoplasm, in the membrane, and only rarely a few were in the outer membrane and or secreted proteins. This also shows what kind of function these proteins are involved. Majority is unknown, but most of them are housekeeping proteins. Only very few were associated with virulence factors. Here we generated a list of top 10 proteins, which we want to look more closely to it. And here you see in how many references they appeared, and this goes quickly down. Our own experimental approach was two-dimensional electrophoresis. We used a field strain from a sheep where we generated protein extract, and then we separated by pH and by size. We used Q fever outbreak sera for Western blot, negative sera as negative control, and then we also stained by Comasi. And then we did an overlay and spot matching and picking. And this is one example of a membrane or an overlay. And we could identify two new proteins shown here and here, which were always detected by sera in 3 out of 4 experiments. And the first gene is involved in the fatty acid metabolism, metabolism, and the second one is part of the glycolysis. So all these both are housekeeping proteins. Then what are we doing? How to predict B-cell epitopes? B-cell epitopes have characteristics that they are part of the proteins which are surface exposed. They are mobile and flexible parts of the protein, and they do not have a 3D conformation. And this you can detect in a protein by the disorder tendency plot, which we used. So what you do is you have your protein, which you identify maybe from the literature or from your own experimental approach, and you align it with all available protein sequences from Coxiella and certain other bacteria, which in the case of Coxiella are also abortive pathogens like Brucella or chlamydia or kind of bacteria. And the alignments are here shown for one not conserved protein and for one highly conserved protein throughout the bacterial world. This is an outer membrane protein, and you pick areas of the protein where you have a disorder tendency above 0.5. And from this area, you generate small peptide fragments. And here for DNA-K, which is a chaperone, which almost every bacterium has, you see in this area, we choose peptides which were highly similar for Coxiella species, and but different to other bacteria. And then you test these peptides by ELISA, and this is shown here for some of our peptides. Here you have serum pools from positive herds, sheep herds. These were different outbreaks. And then you have negative sheep herds, serum pools from negative sheep herds. This is also positive and also from vaccinated animals. And every peptide you test with every pool, and after the pools you test against every single serum. And we could find here in this example 3 peptides which reacted at least with 3 out of the 4 positive serum pools we used. They did not react with negative sera, and 1 or 2 also reacted with vaccinated animals. So these are good candidates for developing a peptide array, but it's not enough so far. So at the end, our advantage is that we use field strains, that we use field sera, positive and negative. And so far we have 17 peptides, which are promising candidates. And there's one other project we have. Instead of using small peptides, we use recombinant proteins, and we have done this for one. The COM1, it's the outer membrane protein 1 from Coxiella. It's also a chaperone and was also one of our top candidates for as immunogenic protein from the literature. So we cloned and purified this, and we tested the same serum panel we tested for the peptides. But here with the ROC curve analysis, we found sensitivity and specificity for all 3 ruminant species very low, from 71 to 90, or only from 67 to 76. So one recombinant protein alone is not enough. Promising, but not enough. And this is all. Thanks, and I'm happy to take any questions.
Thank you, Dr. Katja. Any question from the participants, please? Yes, Professor Feizabadi.
Hello, thank you very much for the nice presentation. About treatment of Q fever and failure in antibiotic therapy, I would like to have some information about this.
For human patients.
Yes.
So
Yes.
normally you give doxycycline for several weeks. And one thing I forgot, there is no licensed vaccine for humans. Only Australia has one vaccine, but you have to test the people before you give the vaccine because of severe side effects. That's also why it's only allowed in Australia. And for animals, there is a licensed vaccine for sheep and for cattle, but not for, no, for goats and for cattle, but not for sheep.
Okay. What about treatment failure? I mean, drug resistance in tick fever, in Q fever?
For humans, I'm not sure. I know there are some chronic cases. So you have normally 60% of all infections in humans with Q fever are asymptomatic, and 40% will develop acute Q fever. And some of these will develop a chronic disease. So 1 to 5% of infections will go into a chronic disease, and mainly people which have a former heart disease will develop endocarditis with Q fever. And I think this is almost lifelong.
Okay. You know, in our country, we didn't did so much research on Q fever until Dr. Mostafavi started working at postdoc, because we have a very big gap since the last time that we received reports on the occurrence of Q fever in south of Iran. So I would like to invite Dr. Mostafavi and his colleagues at postdoc to brief the audience about the current situation of Q fever in the country and also in the region, if it is possible. Thank you.
Yes, thank you, Dr. Faisabadi. As you know, we in Pasteur Institute of Iran are the national reference laboratory for Q fever. Mm-hmm. And lots of human and animal samples are collected from different areas of the country. In recent years, we have found lots of both acute and chronic forms of the disease in samples from different areas. We have found some positive endocarditis samples from Tehran, from Mashhad, from Kurdistan, which are from different areas of the country. And also we have collected samples from the ticks from the north and from the south, east and west of the country. And we have found also Coxiella in collected ticks. So it seems that, and also samples from the animals, the milk and the other samples, who my colleague, Dr. Saber Ismaili, who is involved directly to this project, we have found lots of positive samples from both human and animals, and lots of them are published and are available to be reviewed.
Mm-hmm. So the samples mean you have DNA, or you have the bacterial, you isolated the bacteria?
No, we have DNA now.
Mm-hmm. So it would be interesting to type these bacteria to see if you also can see a tendency in genotypes and yeah, to see if genotypes are corresponding to chronic and acute.
Yes, it is followed, and it is one of the presentation of Dr. Saber Ismaili in the next panel 2 weeks later. He will discuss in details about what we did for genotyping of the Coxiella samples from both human ticks and animal samples.
That would be very interesting for me, so maybe you can invite me.
Yes, of course. Thank you.
Yep.
So you believe that a lot of cases now have been neglected in the country with Q fever?
Yes, yes. It is a real neglected disease.
Yeah, you should maybe do a broad overview or surveillance of the animals. And you mentioned ticks. You know, there's a hot discussion about the role of ticks in Coxiella. Because like in Germany, we have 2 tick species which are dominant. And we found almost none of these ticks carry Coxiella. But then if you go further south, other tick species carry Coxiella. And then there are also Coxiella-like bacteria where the PCR cross reacts. So this is a really interesting topic to see what Coxiella do you have in there.
Yes.
Dear Katja, have a question.
Yes,
I have
Saber, please.
Thank you for accepting our invitation. We are very happy that you are with us in this Congress. Your lectures was very interesting. I have a question about what is your suggestion for me, molecular method for the differentiation Coxiella from Coxiella-like bacteria or in the symbiontic bacteria, especially in the study in ticks.
Yeah.
Also, we need to differentiation from Coxiella to Coxiella-like before genotyping, MST genotyping or other methods. Some genes included 16S are not suitable for this purpose. What's your suggestion?
I would suggest to use IS1111 and sequencing of it. You can sequence a transposable element and then you can differentiate based on the sequence information. But I can also send you a paper from Raoul, you know Didier Raoul from France. I think I read his method now twice. He uses 2 different, I have not in mind now, 2 different approaches, and I think it's very smart. So I can forward you this paper.
Okay, thank you.
Mm-hmm.
Thank you.
You're welcome.
Okay, if there is no more question, and if you agree, we can go to the last lecture. Before going to the last lecture, I would like to thank Dr. Ismaili, Saber Ismaili, who was one of the main persons who followed arranging this panel and inviting our colleagues for this meeting. The last lecture will be, it is not another neglected disease. It is about COVID-19, which is a pandemic disease nowadays. It is presented by Professor Mohammad Mehdi Gouya, who is the director of Iranian CDC. As he was not available at this time, he is recorded his presentation, and I will share with you his lecture now. Before going to this lecture, I would like to thank Dr. Katja again for her nice presentation. Thank you, Katja. Okay. Let me share, sorry. Okay.
In the name of God. First of all, I would like to thank microbiology Congress committee for inviting me for this important Congress, and thank all of them for their great job. Next slide, please. Let me give you some information about our important measures taken during the pandemic, before pandemic, during. Another activity we have done during this time had been development of rapid response teams at provincial and national before the pandemic, and training courses for them with support of WHO before pandemic and during pandemic, field investigation programs and monitorings the first suspected COVID-19 signals. Next activity had been monitoring and prevention and control, case management and data management for this issue. And we have provided non-pharmaceutical interventions also, and medications, vaccines, and PPE supplies. Next slide. We have utilized of available strategic supply of medical countermeasures before the beginning of the pandemic. And for us, one of the most important issues had been assistance to national reference health laboratory and advanced laboratories at national and provincial level also. Next slide. Strengthening collaboration with neighboring countries and international partners was very important for us. Public and professional risk communication and community engagement also was very important, and we have done a lot in these areas. Early warning system for us was very important. Based on this issue, we decided to think about syndromic surveillance system and establishment of this surveillance system at national and some provincial levels as a pilot project regarding COVID-19 and enhance evidence-based monitoring programs. Next slide, please. For point of entries, we have had point of entry surveillance system from several years ago, but improvement the quality and quantity of this surveillance system, and also capacity building for the persons who are working in these areas, and monitoring and evaluation of national and provincial performances for COVID-19 pandemic. Next slide, please. We think something has been very important for us. We should consider them, their strengths and also opportunities to improve these weaknesses also. Political will and commitment for control of COVID-19 was very important issue, and I think for the next steps of pandemic also will be very important. Engagement of Ministry of Health in multidisciplinary coordination for uniformity for COVID-19 pandemic control also, and enhance laboratory surveillance for detection and follow-up, also case management and contact tracing, and providing different guidelines in different areas of control program for the pandemic. Next slide, please. Second version of syndromic surveillance electronic enterprise at countrywide have been one of the further actions, also updating the COVID-19 prevention and control program based on the evidences and strengthening risk communication and community engagement for the public and professionals, and improving infrastructure for combating public health events, not only for COVID-19. Preparation and strengthening laboratories for detecting variants of concern, variants of interest, and variants of highly consequences was very important for us, and we think this is one of the most important activities for us to do it as soon as possible also, and expanding joint and national vaccine RCTs and production of COVID-19 vaccines through joint ventures and domestic companies. Next slide, please. Let me give you some information regarding our actions taken to address vaccine need of the country. We think from the first step of pandemic preparedness for vaccine production in three different areas: domestic production, providing vaccine through COVAX facility, and bilateral agreement also. Next slide, please. For domestic production, Islamic Republic of Iran is actively following the development of 4 types of vaccine platforms. One locally produced vaccine, which is inactivated, has received EUA at national level approval. The other platform, which is produced jointly and is conjugated recombinant protein subunit, has also received EUA at country level. Also, adenovirus and other platforms are under development. Let me give you some information regarding last situation update of COVID-19 vaccine administered in Islamic Republic of Iran till 31st of August 2021. Total number of first dose administered at country level had been more than 18 million. Total number of second dose administered had been 8,700,000, and also total doses administered had been till now more than 27 million. Next slide, please. Thank you very much for everything.
Okay. Thank you again. I would like to thank you again, Dr. Feizabadi, Dr. Abdullahi, and other colleagues who were the hosts of the meeting. It was the last lecture of our panel, Emerging Infectious Diseases panel, and as Dr. Gouya is not available here, we will not have any answers for any possible questions. Dr. Feizabadi, the floor is yours, and if you agree, then we can finish the meeting. Thank you.
Thank you very much for your efforts made for our Congress, in this Congress and other congresses. Also, I would like to thank the invited speakers from France, Germany, and Italy, and I would like to inform them in the future we have more guests from these countries. So thank you very much and see you in our next panels.
Bye.
Thank you. Goodbye.
Bye.
Bye. خداحافظ.